So in part 3 of your Kin 425 class for January 27th, I'm going to go over important aspects of carbohydrate oxidation and fat oxidation. So this continues on from the previous video. Once pyruvate or lactate have entered the mitochondria, which metabolic pathway do they go through? Hopefully you can remember from your Kin 225 that this would be the Krebs cycle, also known as the tricarboxylic acid cycle or the citric acid cycle. So I've got a detailed diagram of this cycle, and there's a lot of metabolites and enzymes that are involved in this cycle that you don't have to know for this course. That would be restricted to a detailed course in biochemistry, but these are the key steps in this pathway. So one thing to keep in mind with this diagram is that when you break down glucose, glucose is a 6-carbon molecule. Pyruvate is a 3-carbon molecule, so you have 2 pyruvates that get formed, and so you have this cycle going around twice for every glucose that you break down. So you can see the key steps here is you have pyruvate being converted to acetyl-CoA. This is a 3-carbon molecule. This is a 2-carbon molecule. You're losing carbon dioxide here, and that's part of the carbon dioxide that you breathe off during exercise. A key point here is you also have formation of NADH. And then as you go through this cycle, again, you give off carbon dioxide. So that's where you lose those other 2 carbons from acetyl-CoA. Again, that's expired in your breath. And at different points in this pathway, you have NADH that is formed, and you also have FADH. The importance of forming these molecules is these molecules eventually go to another aerobic pathway in the mitochondria called the electron transport chain. For every NADH that goes to the electron transport chain, you get production of 3 ATPs, and for every FADH that you have going to the electron transport chain, you have production of 2 ATPs. So you can get a lot of ATP from breakdown of glucose. This pathway goes around twice, so you got to think that everything gets multiplied by 2 here. You get 1 ATP that's produced directly in one cycle of this pathway. So again, it goes around twice. So you get 2 ATPs from one molecule of glucose. Key enzymes that are used as markers of this metabolic pathway include citrate synthase and succinate dehydrogenase. So if you happen to be working in a biochemistry lab where you're assessing muscle biopsies, you can assess these enzymes quite easily, and those serve as markers of aerobic metabolism because they're markers of this metabolic pathway. So this metabolic pathway involves a high number of enzymes, but these are the ones that are assessed most often.